The second part of the human variation lecture series, Module 5, is focusing on Clines. And a Cline is just a gradient of genotypes across geographic space. And we can sample a number of human population groups on large continents, and we can show that distribution over this geographic space, and we can try and answer some questions as to the origin of that diversity. What evolutionary mechanism actually created that diversity? And I have a few different examples. The ABO blood group, which we've worked on, sickle cell trait, which has already been mentioned. And we'll go into more detail in other topics like lactase persistence, as well as skin color or skin pigmentation. As I just mentioned, Clines and clinal variation is just a gradient of genotypes over geographic space. So we oftentimes try to collect data on organisms, I mean human groups, that are native to a specific region. Now, of course, people move around, there's migration. So, for example, if I was talking about Australia, I'd be discussing Aboriginal Australians, not Europeans that moved there afterward. So blood polymorphisms like the ABO blood group are well studied. And when you look at variation, then it would be due to some of these accumulated differences in those specific environments. Here's one of the things about the ABO blood group and other blood polymorphisms. We're not quite sure if there's really any adaptive significance. We know that when an organism has all the same trait, like for instance, we all have eyes, two eyes, nose, and a mouth, right? And that's fixed by natural selection. We all have those features. There's no real variation. Somebody doesn't have a nose or not. And when we have variation in things, that means that natural selection hasn't acted on it, right? And so it's likely something that came through in a non-adaptive way. But you do see some patterns, some general patterns in the ABO blood group when you compare major continental groups. One is that the O phenotype is very common among American Indians or people who are native to the Americas, both North and South, matter of fact. B is common in Eurasia, and A is common among Aboriginal Australians. Here's one example of a Cline being able to sample a blood type or a genotype and disperse it over geographic space, and then try to come up with a few ideas as to why we observe this variation. So in this chart, the areas of blue are designated as 90 to 100% of O blood type. And then you can see the varying degrees, 80 to 90, 70 to 80, all the way into things like orange and bright yellow that are 60 to 750, 60 or less. You can notice that virtually all of native South American people are O blood type. And you can see it's predominates in Central America, in the American Southwest, in parts of Florida and the Caribbean, and then areas in parts of modern day Alaska and Canada. And it's 70 to 80, 80 to 90 in the rest, for the most part of North America. So what happened? Well, remember that to have O blood type as a phenotype, it's recessive. So you need to have two copies. What it likely looks like is just random drift, probably the bottleneck effect. Think about it. If you have smaller land to traverse, you're going to have likely less people traversing through it. And that will create a bottleneck effect where you will actually see less diversity as you move further and further away from a population origin point. So the way this kind of looks like is that we know that human evolution occurred in Africa. And as you move away from Africa in terms of a distance, we can kind of predict your genetic diversity. As you move further and further away from Africa, there's less and less genetic diversity. And so this makes sense. If most people in the Americas are homozygous recessive, meaning they have all the same genotype and phenotype, It's likely because a small number of individuals founded the populations in the Americas. We think people moved from Siberia into parts of modern day Alaska or along the coast and populated the Americas. Think about that. A journey where people migrate from a larger gene pool, and the smaller gene pool contributes largely to the daughter population. Sickle cell trait is an example that we have already went over. If you have sickle cell trait, that means you are heterozygous. You have one copy of the normal hemoglobin, and you have one copy of the sickle cell allele. And we actually notice the sickle cell allele increase in some populations. Think about this. The assumption is that a disease allele will be removed or deleted from a population by natural selection. Natural selection keeps good things there and it removes bad things. So the obvious, mechanism that natural selection would implement with a disease allele would be to decrease it or remove it. But why is it increases? Why is it increasing? Well, people that have sickle cell disease and sickle cell trait, they confer some type of immunity to malaria. And what this means is if you have sickle cell disease, you won't get malaria, but you have a disease where you have some major health problems with being able to transport oxygen to the rest of your body, and you are highly susceptible to iron deficiency anemia. Now, if you have two copies of normal hemoglobin, you are homozygous dominant, then you don't have any health problems really associated with you because you don't have sickle cell trait or disease, but you're highly susceptible to malaria in any of these endemic areas. That's why it's important to be heterozygous in this region. And matter of fact, heterozygous advantage would mean that somebody's heterozygous for this trait, that they probably have the most preferred genotype in any human environment, particularly ones that malaria is endemic. Lactase persistence is always a neat topic to discuss. I use this term lactase persistence because it helps explain that when you were a child and you were able to digest your mother's milk, how can you not into adulthood? Well, when you are a baby, most people produce lactase, which allows you to digest the protein lactose in, for example, the milk of a cow or the milk from your mother. Some individuals continue to produce it after adulthood, lactase persistence. That means that if you do continue to produce lactase into adulthood, you are lactose tolerant. If you do not continue to produce lactase into adulthood, you are lactose intolerant. On this chart, I am showing you the clinal variation of lactase persistence. In the very bright green areas, you have the groups that have been sampled that are the most lactose tolerant. In the red, the brown and the red and brighter red, you have the groups that were sampled that are the most lactose intolerant in terms of percentage. You'll notice that people assume that this was a black and white issue in the past, that all of Africa and people from African descent, that they're going to be mostly intolerant. Well, you can see there's diversity in Africa. Most of sub-Saharan Africa and the Democratic Republic of Congo are tolerant, but at areas in South Africa, they are not. You can see the People's Republic of China, for the most part, in areas of Southeast Asia and Japan, mostly intolerant. And this is because they don't really drink milk on a grander scale, because that's typically seen as a Western diet. So you can see in parts of Russia, in parts of Europe, as well as the United States and Australia. Now, you can see in Australia, it's bright green because that's looking at it as an entirety, because the majority of Australian people have European ancestry. That's we think it's green because of people that migrated from Europe, and Aboriginal Australians are mostly intolerant. That's why you have a little red figure. If you look at the United States, for the majority is lactose intolerant, but there are certain groups like African Americans as well as American Indians that migrated from places that didn't necessarily adopt drinking milk, and of course the majority of American Indians, they did not adapt this type of pastoral life either. So you can see that we showed clinal variation, and we were able to discuss a number of different evolutionary mechanisms, like migration, that explains the variation in, for instance, like Australia. Skin color is always a really interesting topic to think about in terms of human evolution as well as human variation. We know that solar radiation is important for us to produce vitamin D. And so the sun's rays, and it may not even take that long, allows us to produce vitamin D, which is really important for our bone growth. If we cannot synthesize vitamin D, we're not able to actually use calcium in our bodies. And this is bad for our bones and makes them weak and causes abnormal growth. And we do know that if you have prolonged solar radiation, then you actually can potentially get skin cancer. And solar radiation also depletes a B vitamin known as folic acid or folate. And so if you think about it, the sun's rays, we need it for vitamin D, but it can cause skin cancer as well as deplete B vitamins. So that would mean if you have a darker skin pigmentation, that your ancestors evolved in an area where it was an advantage for darker pigmentation. And so that would mean typically closer to the equator at the extents of the Tropic of Cancer and Capricorn with a varying degree. And that would also mean that lighter skin is better at higher latitudes because then you have lighter skin to absorb sun that may not necessarily be there. If you have ever gone or know anything about England, the sun shines like once a month. So we don't need a lot of exposure to get vitamin D. And if you think about very little exposure in high latitudes, but when the sun is out, right, the skin is light enough to get that exposure, to get the necessary vitamin D. This also means that individuals that are on the opposite in different environments, they are at a disadvantage. So that means that people with darker pigmentation or increased pigmentation, it's a disadvantage at high latitudes. And it's a disadvantage for people with lighter skin or lighter pigmentation closer to the equator. So somebody like me who has lighter pigmentation, living very close to the equator in Africa, which I have done, my light skin is a disadvantage because I'm highly susceptible to skin cancer and I'm highly susceptible to my folate reservoirs being obliterated. And you can see this in African Americans in the northern U.S. because if an African American who lives in, say, maybe Minnesota or Wisconsin, right, they have to stay indoors because it's so cold. They're not getting outside and getting a lot of exposure to sunlight and vitamin D. Now, this is the same instance in parts of East Indians, in parts of Great Britain with East Indians and Pakistani people or people from Indo-Pakistan. So this isn't much of a problem anymore because a lot of our food is fortified. You grab a carton of milk here, it is fortified with vitamins A and D, and in many of our cereals and foods are fortified for this very reason. If we were expecting a 1-to-1 ratio to the solar rays that are absorbed in each one of these regions of the world, we should see a chart that looks like almost like this, right? In the hottest areas, we would predict to be the darkest pigmentation. But when we look at the clinal variation in skin pigmentation, the picture is slightly different. So in this chart, you can see individuals with the darkest pigmentation are all centered in pockets of Africa, but you will also see darker pigmentation across parts of Arabian Peninsula, India, Southeast Asia, and Australia. As you move away from Africa, you can see lighter and lighter skin color in parts of Eurasia as well as the Americas. If you noticed, there were pockets of the Southwest that had very, very intense pigmentation. And this is interesting to think about. If people from the Americas originated in the Americas at the same time as people in Africa did, hundreds of thousands of years ago, you would likely see the same amount of pigmentation. But we think that the populations in the Americas are much younger, that they hadn't accumulated as much time as people in Africa to have this much more increased pigmentation in comparison.